Dr. Zitnik is an assistant professor in the Department of Biomedical Informatics at Harvard Medical School, associate faculty at the Kemper Institute for the Study of Natural and Artificial at Harvard University. She's also associated member at the Broad Institute of MIT and Harvard and affiliated faculty at the Harvard Data Science Initiative. So overall, Dr. Zitnik investigates foundations of AI to enhance scientific discovery and to realize individualized diagnosis and treatment. The overarching goal of her lab is to lay the foundations for AI that contribute to the scientific understanding of therapeutic design and genomic medicine. So her lab is pioneering AI systems informed by geometry, structure, and symmetry, grounded in scientific knowledge. And I think she's going to talk a bit about her latest work. So thank you for joining us today, and the floor is yours.
Of course. Good morning. I'm excited to be here with you today. I hope you can hear me well. So I will discuss some of our recent work on how we can shorten diagnostic and therapeutic odysseys for patients that are very difficult to diagnose. And throughout the presentation, I will highlight several projects. They're all linked from our lab website together with associated code bases, data sets, tutorials, examples of usage, so you are more than welcome to check them out. So over the last several years, there have been considerable advances in AI. That's not the audience that's not aware of that. And I see that there are these two key impacts that advances in AI, particularly through foundation models and large-scale pre-trained models, have had on medical research as well as translational research. On one end, these new types of tools can enhance and improve or have an impact on how we go about our everyday research activities. And on the one hand, an important consideration is that these models can start providing insights that might not have been easily achievable using traditional models just a few years ago. And those might be insights that are provided at temporal scales, at spatial scales, or in the long-tail distribution of diseases or patients that were not accessible, easily amenable to machine learning analysis a few years ago. And so we are seeing this evolving use of data-driven models for several decades, and now I believe we are in the stage of interactive foundation learning models. We're slowly transitioning towards various agents being built around operationalizing LLMs and related foundation models to be more compound systems that are compatible with real needs in medical research and everyday activities. But with that in perspective, what I would love to discuss with you today is this notion of developing AI models that can really help those patients that are hard or difficult to diagnose, hard and difficult to treat for frontline clinicians in particular. So this presentation will have two parts. In the first part, I will outline Shepherd, which is our model for diagnosing patients with suspected rare genetic diseases. And the second part of the talk, I will discuss TxGNN, which is our model for drug repurposing in a zero-shot manner across many diseases that currently have no or very few treatment options. And the overall motivation for this work is to shorten both diagnostic odysseys that rare disease patients have to go through, as well as then therapeutic odysseys that those patients have to go through once they are eventually diagnosed. And we are seeing that the opportunities of these high-capacity models in these areas can be incredible if we can make these models really helpful and useful for clinical researchers that deal with these patients. So what are diagnostic and therapeutic odysseys in the context of rare diseases? There are over 7,000 rare diseases worldwide. They all collectively affect hundreds of millions of individuals. But every rare disease, as the name implies, has a relatively low prevalence, and there are a small number of patients associated with it. But because there are so many rare diseases, collectively rare diseases are common, so they affect a large number of patients. The problem with rare diseases and patients suspected to have rare diseases is that they are often very challenging to diagnose. Frontline clinicians might not have encountered many patients with that specific phenotypes or presentations during training. And so those patients then typically go through several years of search, which is this odyssey process, to finally eventually reach the correct diagnosis. However, during that time, one of the challenges is that their disease might progress in a way that's not easily reversible, as well as they might be exposed to various other issues. And developing treatments for those diseases is also incredibly challenging. Why? For one of big challenges is that large pharmaceutical industry is simply not incentivized towards developing drugs for those diseases because patient populations are so small. And the cost of the drug development process is much, much larger than what the downstream profit might be if the drug exists. And second, many of these diseases are very poorly understood both at the molecular and the clinical level, which makes the development process even more challenging. So you can see that there are all these challenges that arise with scarce label data, incomplete knowledge in the context of rare diseases that we would like to address in a meaningful way towards the models. So let me first talk about diagnostic odysseys. So the motivation for Shepherd here is that the current statistics in the United States is that for patients with rare diseases, they go through on average 5 to 7 years of the search process before they arrive at the correct diagnosis. During that time, they visit many doctors, many specialties. They are on average misdiagnosed several times, and they need to go through a process of referrals. And so really the challenge is that this diagnostic delay is so pervasive that creates a big real problem for patients. A nationwide initiative in the United States that deals with this problem, it's called UDN, which stands for Undiagnosed Diseases Network. This is a nationwide program with 12 clinical sites. And patients that are difficult to diagnose can eventually be referred to the Undiagnosed Diseases Network program. Within that program, those patient data are managed by a dedicated clinical team. Their genomes are sequenced through exome sequencing or full genome sequence. They go through a detailed clinical workup, and that's a very expensive, tedious process that aims to eventually diagnose those patients that cannot be diagnosed otherwise. And so our overall goal was to develop Shepherd model that can provide insights into what might be the correct diagnostic label for patients that somehow their genetic makeup plus their phenotypes do not match easily to any of well-established disease in a medical vocabulary. So fundamentally what this means is that we are dealing with this problem of AI-based medical diagnosis. Those who are here in the audience know that this is not a new problem. There are so many algorithms out there, traditional machine learning models, deep learning models, recent foundation models that are developing capabilities that can help with medical diagnostics. And I'm just highlighting 3 recently published examples that include very famous and incredibly instrumental work early on in developing models that can help with diagnosing diabetic retinopathy by training large deep learning models on hundreds of thousands of images, models that go through large number of electronic medical records for pediatric patients to help with diagnosing common childhood diseases, models that can help with diagnosing skin lesions, whether they are benign or malign, based on their semantic representation. And what we argue, however, is that, well, none of those models apply to rare diseases. So the challenge with rare diseases are just fundamentally different than the challenges with diagnosing other diseases such as cancers or common childhood diseases. And the fundamental problem is that data sets, meaning the number of patients with labeled diagnoses per each disease, are so small that we can simply not afford to train some of more like usual deep learning architecture for this problem. And this problem is such that we cannot solve it easily by just going out and try to find more patients and try to label those patients. There are no more patients to be found. And so what we need is then a completely different approach that would allow us to be able to leverage very heterogeneous population of patient data, where for each disease we have a small number, a handful, of labeled patients per disease, or perhaps no labeled patients at all. And what can we do in that context? And this is the problem that Shepherd was really motivated by. So what is Shepherd? Shepherd is a few-shot predictor that is focused on hard-to-diagnose patients. The way we design and think of Shepherd at a very high level, you can think of a model that has 2 inputs. For a given patient, the first input is a list of variants and associated genes that the patient has mutations in. So this is generally an output of a variant prioritization calling algorithm. The second input is a set of phenotypes that can come in the form of human phenotype ontologies or symptoms assigned to the patients through the clinical workup. So given these two inputs, then Shepherd model will assign and align those two inputs onto a medical knowledge graph to contextualize the genetic and clinical information about that patient with the ultimate goal of performing three key capabilities. The first one is that of causal gene discovery, where the task is really to produce a ranked list of genes as the output, such that hopefully the genes that are associated with the disease or cause the disease, if this is indeed rare genetic disease, would be ranked very close to the top of the list. The second important capability is what we call patients-like-me functionality, which is that of taking the patient's data and find in a large healthcare database, are there other patients that share similar clinical or genetic features that can be looked at and reasoned about in the context of what worked perhaps well for other patients or what didn't work for other patients. And the third important capability is what we refer to as novel disease characterization. Many of patients that are part of this program have novel, unusual representation of a known rare disease, or sometimes they have mutation in a gene that has never been previously associated with the disease, or they have a novel mutation in a previously disease-associated gene. So things can become very complicated quite quickly, and so that means often that it's not easy to take a patient data and map them reliably to a single diagnostic label, like MeSH ID or disease code, but instead what we provide is then an information about how a patient can be mapped to a distribution of disease phenotypes. So it's the model says, like, it looks like 20% of one disease, 5% of second disease, but there's no easy way to match the patient to any individual disease based on the data that they have. Okay, so how do we train this model? So we train this model in two key stages. So the model is first pre-trained in a patient-agnostic manner, where the goal of the first stage of pre-training is to learn a general encoder of medical knowledge that can capture well current known relationships between diseases, between genes and diseases, between variants in those genes and genes based on existing knowledge about rare diseases. This is then followed up by what we call patient-centric training, where the model is additionally pre-trained on patient data with the twist that what we train the model on here are simulated or synthetic patients. And finally, after that pre-training stage, we take the model and we can fine-tune it using small amounts of real-world patient data, or we directly use it for predictions, such as patients-like-me retrieval. First step, by patient-agnostic training is done using a medical knowledge graph. And so specifically for this stage, we're training the model on prime KG knowledge graph, which is a knowledge graph my group has built and publicly released. One of the advantages of these knowledge graphs is that we have a semi-automatic way to update knowledge graphs over time as new data become available. That was indeed motivated by one of key problems of many knowledge graphs that are out there, that are all exciting and wonderful in their own way, but oftentimes when they are published, then nobody updates them later on, so they become obsolete quite quickly. And it's easy for us to update, and we do release new versions of knowledge graphs roughly every 3 months. All are available from the project website. And the second important consideration is that we wanted to design this knowledge graph such that it has a really broad representation of disease phenotypes. Many medical knowledge graphs out there are deeply focused on one or a handful of diseases. And so that did not work well for us because we want to have a broad representation of diseases and disease phenotypes. So in that regard, its unique features is that it contains over 17,000 clinically recognized phenotypes. Okay, so once that step one of the training process is performed, which is patient-agnostic pre-training on the prime KG knowledge graph, where the objective is to generate embeddings or latent representation of each and every node in that knowledge graph in such a way that two concepts or nodes in the knowledge graph with similar local connectivity neighborhoods will be embedded close together in the embedding space, and this is achieved through a form of heterogeneous graph transformer model. Then we proceed to the second stage, which is that we continue to train the model, but now in a patient-centric manner. And so for that, what we have is a pipeline that can generate realistic rare disease patients. I could have a separate presentation just on that. Happy to take questions around this in the discussion point. But that pipeline allows us to generate patients that look like real-world rare disease patients. And for a large cohort of those synthetic patients, we overlay them on the knowledge graph by defining subgraphs of genes and phenotypes that each of those simulated rare disease patients would have. And then in the patient-centric training, we embed those subgraphs of the knowledge graph that represents patients. And so ultimately what that gives us, it gives us representations or embeddings not only of medical concepts in the knowledge graph, but also patient embeddings, which are these subgraph embeddings. So the three key features of Shepherd algorithm that makes them suitable for rare disease diagnosis are then the following. So one is we are found a reasonably well working way to train the model on simulated data, and simulated patient data. I think what's most exciting is that we evaluate the model after the model has not seen any real-world patient. So that was quite striking and not surprising to me that it could work in any way. The positive side effect of that is that our model is publicly available. It was very easy for us to share it across institutions. We are now running several pilots internationally because it was so easy for my students to really just, after 1 or 2 Zoom calls with collaborators from an external institution outside of the US, share the model, share the scripts, and tell the collaborators how can they use the model directly or potentially additionally fine-tune it using their local private data. The second important consideration is that we model patients as subgraphs in the knowledge graph. And so why this is so important is because we often see that for these challenging patient cases, if you look at 2 patients, A and B, and look at their variants and look at their phenotypes, there will be zero overlap, direct overlap in genes, in variants, and zero overlap in phenotypes. And so based on that, we would naively conclude that these 2 patients are completely unrelated to each other. But when you look at them closely, is that, well, they might have variants in the same genomic regions, or their phenotypes might not be directly verbatim the same, but they are linked indirectly through a phenotype ontology. So when there are no direct overlaps between patients, but these indirect associations exist, the subgraph-based representation can capture there because we can model proximity between patients in the knowledge graph. And the third important component is that of being able to perform label-efficient training that for a small number of patients that are simulated patients, we can still train the model well. Okay, so once the model is trained, just to recap, there are 3 types of diagnostic tasks that we centered our evaluations for that I showed and briefly discussed earlier that include causal gene discovery or disease gene prioritization, patients-like-me retrieval, and third, characterization of novel diseases. And that was in part motivated by our close collaboration and discussions that we had with clinical researchers in the Undiagnosed Diseases Network in terms of what would be the kind of questions they would like to pose to the AI models when they work with patient data and these UDN patients across different stages of diagnostic workflows. So to recap how the experimental setup looks like, so we train Shepherd model on 42,000 synthetic patients using this generative pipeline that we developed. After the model is trained, then we can, pre-trained, then we can use it in 2 different regimes. The first regime is to fine-tune it on small amounts of local patient data, and then take the fine-tuned model and evaluate it. The second regime is to do no additional fine-tuning, but just evaluate the model directly. For the experiments I will show in the next few slides, we evaluated the model on 3 external cohorts of difficult-to-diagnose patients. The first one was the UDN cohort, which is a cohort of 465 rare disease patients whose diseases span 299 diseases. So you can see here simply there's really like 1 to a handful of patients for each disease. The second one is my Gene 2 cohort, and the third one is the Decipher cohort from the UK. Okay, so let me show some of the plots here. So we did lots of the usual benchmarking as you would expect. What this slide shows, it shows performance of the model in terms of how highly it has ranked the gene that was ultimately shown as disease-causing gene, which is often determined through additional functional animal studies. But we were quite confident in those labels that were provided by clinical experts. And so what we can see is that Shepherd has an average recall when we look at top 5 genes that the model is asked to be ranked, we're around 75%, which means that, the disease-causing gene was ranked very highly by the model. And so we compare that to a suite of traditional approaches or network science models, and also to some of the domain-specific approaches, in particular variant prioritization calling algorithms that are currently used in practice within the UDN. The part that I'm actually more excited about is this slide, which is the slide that shows performance and improvement of the model when we look at the most difficult patients to diagnose. And so here is, for an example, is an improvement of Shepherd over High-5, which was the strongest performing baseline model that considers both phenotypes and genetic variants to identify disease-causing genes. And we stratify model performance by the nature of patients' ultimate diagnosis. And there are some patients, for example, that will ultimately diagnosed with the disease based on the mutation that they had that was never seen before in any real-world patient. So it was a new mutation in an existing disease-causing gene, or a new mutation in a new, not previously known disease-causing genes. And so those are particularly hard cases for existing models to pinpoint, and we believe that our current preliminary results shows that the reason why we are better at this type of patients is because of the ability to model indirect associations through the knowledge graph. We can answer lots of these patients like me type of queries. So what I'm showing on this slide in the central part is this latent embedding space of 42,000 synthetic patients. Every dot is a patient. They are colored by the disease category that they were sampled from. And on top of this embedding space, then we can embed real-world patients. And shown are 4 real-world patients, which are patients from the UDN cohort that were ultimately diagnosed with the gene that the model also correctly predicted well. And we are seeing that those patients that were real-world patients embed in this latent space in the neighborhood where lots of similar synthetic patients are found. And we are particularly interested in this task because what we are currently running as a follow-up study is working with a large healthcare system using this algorithm to scan electronic health records of millions of individuals to potentially find what are those patients that might have been misdiagnosed that could benefit from being recruited into some of more deeper, very precise, but also costly precision medicine clinics such as UDN. And third is characterization capability of the model is that of characterization of novel diseases where the task is to look at what are the disease embeddings that are found nearby patient embeddings, especially for patients that cannot easily be assigned to any individual disease. And so why is this so exciting and important is because for many patients in UDN, what we see is that if you would look at their genetic data and the variants they have, you would predict that they have disease A. If you look at their phenotypes, you would predict that they have disease B, and A and B are very different from each other. And so what can we do then? And so it really becomes challenging and not surprising, because if those patients would somehow align well with existing medical vocabularies, they would not be, they would be diagnosed easily by clinicians and would not go through this extensive diagnostic odyssey process or be part of the UDN. And so what this capability of the model allows us is to characterize the local neighborhoods of patient embeddings in the latent space and determine the distances between patient embedding and embeddings of various diseases. And in doing derive descriptions of potential disease that the patient might have if the patient cannot be easily matched to any disease based on its known knowledge. So just to summarize this part here, Shepherd is unique in a way that it overcomes some of the limitations of supervised learning, also some limitations of self-supervised learning as in the context of rare diseases that do not represent real challenges in other diseases with higher prevalence rates. It can generalize well to novel phenotypes, genes, and diseases. We have numerous pilots at several institutions that we are running now that show strong capability of the model. And more broadly, it has some implications for various medical problems where the data availability and data labels are incredibly or extremely scarce when we're really thinking about a handful of labels per class or patient labels per class. And so because of that, it's kind of lays and paves the way towards better, potentially deep learning powered models for diagnosing patients with rare genetic diseases. When those patients are diagnosed, then the next big question becomes, what can we do for those patients to manage their disease, to cure their disease? And so the question becomes that of shortening the therapeutic odyssey. And so effectively, it means that once the patient is diagnosed, we would like to match those patients to therapeutic benefits. And so that effectively means that what we need to think about is a way for take a patient data and match them to novel compound in development or to existing drug on the market. In the context of rare diseases, the main therapeutic modalities are generally gene editing, which works for some patients depending on where the disease-causing genes are identified on the genome. So I'll defer that to a separate discussion. But the second important modality is that of drug repurposing. And the idea here is that we look at drugs that are already on the market, that we see them on our pharmacy shelves, and see if any of those drugs would potentially have beneficial effects for the patients that we're looking at. And why would we want to do that for rare diseases? Because A, the development costs for drug repurposing are much less than that of developing drugs from scratch, orders of magnitude less, and that the development process is also much shorter. So what we have developed is a model called TxGNN, which stands for an all-disease drug repurposing model. It's a model that is trained on multimodal biomedical data that spans biological scales. And once the model is trained, it can be adapted towards an array of tasks. In this case, the tasks that we are interested in are tasks that predict the likelihood of a drug to be indicated, contraindicated for a specific disease phenotype. So this is not the first drug repurposing model. It's a drug repurposing as a problem of matching existing drugs that are already approved to diseases and expanding their use has been well known in machine learning field, and there's quite a number of models that work and tackle this specific problem. However, what we were specifically interested in this context in this study is to have a model that can perform well for diseases that have very few treatment options. So most of existing models of drug repurposing make the following assumption. So they say, here's my drug is already approved, so I know I will frame it this differently. Here's a disease. For this disease, we already have some existing drugs, and so let me find other drugs that might work for this disease. That's the general approach that existing drug repurposing models operate on. And so the way they make predictions is generally to find some way to score or learn similarity between already known drugs for a disease and this candidate drug. And if this similarity across some level of biological data is high, they would predict that this candidate drug might work well. So what that means is that, well, if I give you a disease for which there are no existing drugs, it fails. It fails quite miserably because there is no example of success already for that disease that we could use to score the candidate drug against. And so the problem becomes even more challenging when we are interested about drug repurposing for a disease with limited molecular clinical understanding, which is effectively what is the case for most of rare diseases. So in this project, we specifically want to focus on zero-shot drug repurposing and do so that we can expand the use of drug repurposing models towards diseases that have very few or no existing drugs, no treatment options, and address the long-tail distribution of diseases. So the way you can think of TxGNN model at the very high level, you can think of it in the way that the model is, again, it's a knowledge-enhanced, knowledge graph-based model that takes as input a modified version of prime KG knowledge graph that I briefly mentioned earlier. And then once the model is trained, we can then predict potential associations between diseases and drugs based on likelihood of predicted indications, contraindications, and also we consider off-label use in some manner for at least additional auxiliary tasks. And there is a component of the model called TxGNN explainer that once the model is trained, it can identify what are paths in the knowledge graph that start at the disease node and to the drug node that the model relies on the most when making a particular prediction. And that is done through a form of counterfactual inference where we are removing nodes from the knowledge graph and asking when would the model change the prediction by the most, and that gives us some sense into what are important nodes and edges. Okay, so when the model is trained, it's really, once it's trained, the input is the drug description, the input is a disease name or disease description, is passed and processed by the model, and the output would be this scores plus information about explanations. So this is, I would say, those who follow literature on knowledge-enhanced, knowledge graph, heterogeneous graph transformers, and those type of architectures. It's a fairly established architectural that we use here with one twist, which is this disease pooling module that we introduced here. And so disease pooling module that we introduced here allows us to pool information from diseases that have richer local neighborhoods, denser local neighborhoods in the knowledge graph, so are better understood molecularly, and we can pull that information and transfer it to the disease that we are currently processing, for which the local neighborhood in the knowledge graph is very sparse because the data are limited. And so this disease pooling mechanism, which we show that is through ablation studies, that it's crucial and important for accurate prediction, is the key novelty that addresses some of the challenges with zero-shot drug repurposing. And the rest of it is a form of advanced neural message passing algorithm trained on the knowledge graph. Okay, so once the model is trained, what are some of the tasks that we're evaluating the model on? So specifically, what we're interested in is how well the model could perform on identifying drug candidates for a disease where it has not seen that disease encountered during training time at all. And so based on that, we built two different types of disease splits. So in what I'm showing here is just a summary of those results, where for scenario A, we consider a standard disease split that is used in published literature, where we just take a set of drug-disease pairs, where pairs are defined based on what drug treats a disease based on known FDA indications, and we split them into train set and test set, and that's how we evaluate the model on the test set. And we see that TxGNN performs a bit better than state-of-the-art models, but it's okay, it sounds great. We're not so excited about that part. We're excited really more about scenario B, which is that zero-shot prediction, where the model is asked to produce a ranked list of drugs based on predicted indication scores for a disease that it has never encountered before during training time. And that's where we are seeing a considerable lift in performance. And that's exciting because only 5% of rare diseases have any FDA-approved drugs. So if you can make any useful predictions for a broad range of diseases there's a real impact that can be made. We can design these data splits in slightly more challenging manner. So not only we take one disease and put that disease and all of its drugs into the test set, but we can take an entire disease area. So we can take diseases that are adrenal gland diseases or diseases that are associated with anemia, put all of those diseases and their drugs into the test set, and then check how the model generalizes to a new disease area. And we've done that across several disease areas. So after the benchmarking has been completed, we wanted to also evaluate the model in slightly more real-world settings. So we've done a number of tests. One is the test where we worked with Mount Sinai in New York City, and specifically looked at 1.2 million electronic medical records. And with the underlying hypothesis that if our novel predictions, so predictions made by the model around drugs and diseases that are not yet FDA approved, if they're of any use and have some potential, we would expect to see them more often than not in off-label prescriptions that clinicians make in that EHR record. And so that's exactly what we are seeing. These two plots that I'm showing in the first row on the far right show what is the enrichment in terms of the log odds ratios when we look at top 1, top 5, top 1 or 5% of ranked drugs for a given disease, And we look at how often those drugs could be used for disease in an off-label prescriptions without being formally indicated for that. So that was one positive side for us. We have also done temporal splits where we look at recent FDA approvals, and specifically we asked the model to make prediction at a specific point in time. We waited for 1 year. There are around 40 new FDA approvals made every year. And then we asked, well, are those new FDA approvals, were they predicted by the model in some way? And predicted ranked highly. And the results were encouraging again. And I would not think this is a gold standard validation because the process of drug development takes many, many years. So it's likely that some of the work identifying during the development phase was part of the knowledge graph, right? But it's another line of evidence that we try to provide here. And finally, we used for now 2 diseases used the model to produce a ranked list of drugs and then ask our experimental collaborators to screen the drugs for these emerging pathogens in the order that the model predicted those drugs, rather than do screening exhaustively. And that led to an order of magnitude improvement in the yield of downstream biological experiments than what would be possible otherwise. After all that work, we were interested in making this tool more broadly available to biomedical community. And so we engaged in what we call clinician-centered design. But effectively what it means is that we recruited a number of human experts who are clinical researchers, pharmacists, as well as drug repurposing experts, and wanted to test and ask them how useful, potentially helpful they would see predictions made by this model. And so this in part was really driven by my long-term observation, where I see that sometimes these ML models, they make correct predictions. So it's like, it's fine to count them +1 towards AUROC and AUPRC, et cetera. But sometimes these predictions are trivial. Human expert would not really need an advanced AI model to make that prediction because it's so obvious to them. And so we wanted to test, what is the level of consensus from the medical knowledge standpoint with our predictions, how useful, potentially useful, those predictions real human experts would find, novel predictions, and also whether these predictions coupled with explanations allow them to better understand and reason about whether a prediction is worthwhile considering further or not at all. And so we designed a tool that is also available online. If you go to TxGNN.org, you should be able to see that allows users to specify diseases, drugs, get predicted ranks, scores, as well as highlighted parts of the knowledge graph, and which provides some mechanism of understanding what the data the model relies on when making predictions. Okay, so with that, I will skip this part and just wrap up here. So in this presentation, I shared with you 2 recent projects in the group, where the first one was focusing on helping patients with rare genetic diseases that are often really in search of their diagnosis through incredibly intensive and tedious process, help them arrive and reach that diagnosis faster and better. And then second is that once those patients are diagnosed, effectively start matching the diseases that they have to therapeutic benefits with the focus on existing drugs. So what we're hoping is that the future for those patients is brighter through some of the models and types of models that are being developed now that could help shorten diagnostic and therapeutic odysseys for those patients. Thank you.